Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My goal, as usual, is to find exceptional people, clinicians, researchers, doctors, etc., and interview them and hopefully ask them good questions where I get information out of them they don't normally provide in, boring articles and, in the news. I want to get to the deep stuff. So today I have Dr. Tony Reid. He's at EpicentRx. He's the chief scientific officer. He has a PhD that was focused on antiviral and anti-tumor activity from Stanford. So we're going to talk about his work at EpicentRx. So Tony, thanks for coming.
Thanks. So just to help you out, the company is it's EpicentRx.
Oh no, I for listeners, my kids make fun of me with my pronunciation, and whenever there's compound words like that or compound things, I mispronounce them. So okay,
everyone does. It's you wouldn't necessarily know, but just
okay.
Just to help out there.
Okay.
And then in terms of my bio, I did my PhD was in biochemistry, but I did study the virus effects on cancer cells, and I did a MD there too. So I did an MD PhD at Stanford.
Oh wow! Okay. And what do you do right now for EpicentRx? What's your work there about?
I'm the president and the chief scientific officer. We developed a platform for engineering viruses to selectively attack and kill cancer cells, and also in the process alter the immune system so that other cancer cells can be seen and treated effectively.
Wow. Are those, those are two very different things. If you may, let's talk first about the virus part. So how do you engineer a virus? Do you have to use cellular machinery to do it, or what's that process look like?
Yeah, so this was a very unique biological experience. So if you think about viruses, let's take the common cold virus. Adenovirus is one of them, and it has about 60 different serotypes. Some of those serotypes will infect the lungs and give you the common cold, but other serotypes will infect the GI tract and give you diarrhea. Other serotypes tend to infect the eye and give you conjunctivitis. So there's a recognition of a selective tissue or a trophism. So certain viruses will target certain tissues like the lungs.
Quick question here. Why do you call it a serotype? I thought sero is blood. Is that an old way or old nomenclature, or what does that mean, the serotype?
Yeah, it's an old nomenclature. It's a way of recognizing it by antibodies that are generated. So you can classify them into serotypes. We sequence them now, and that's why there's so many different types that have slightly different mutations, slightly different genomic sequences in them, all within the same basic structure, but with enough variation that, so for example, some will infect the lungs and cause a common cold, and others will infect the GI tract and cause diarrhea, for example.
Is that, because it's being talked about everywhere, is that what you're seeing with the, the SARS-CoV-2, the coronavirus? Is it doing the same thing, affecting different tissues?
That's right. So you get the coronavirus that infects the lung tissue and does give you bad pneumonia and give you, much more than a common cold. So that some patients actually need to be hospitalized and even intubated. It doesn't, for example, cause other symptoms such as infection of muscle tissue, for example, myositis, or infection of the liver tissue, which would cause a hepatitis. So certain kinds of viruses, for example, will give you hepatitis, and others will give you infection of the lungs, so you get pneumonitis and things like that.
So, all right, back to engineering viruses. You were saying there's all these different serotypes, they affect different tissues. And so again, how do you go about the process of engineering a virus?
It took us several years, but we looked at the molecular switches that let the virus figure out what tissue is the appropriate tissue for it to be replicating in. So we took one that typically infects the lungs. We identified the molecular signal within the virus that lets it recognize that it's in the appropriate host, the lung tissue. And then what we did is knock that molecular signature out so that that particular virus couldn't infect lung tissue anymore. And so it's essentially now a very weakened, you could use it as a vaccine even. It's a very weakened virus that no longer can replicate in lung tissue, and since it doesn't infect other tissues anyway, it's a very weakened or innocuous virus. We can then use that.
A quick question here. How do you, do you culture a large amount of virus and isolate it somehow, and then how would you knock out part of its DNA or RNA?
Yeah, that's a molecular technique. So we go in and sequence the virus so we know exactly what the control sequences are. And then we make very small, deliberate changes in those signaling sequences. And then you have to go back and test them. So you have a hypothesis that a particular signaling sequence is what controls the ability of the virus to replicate in lung tissue. You can then alter that so that switch doesn't work anymore. And then you go back and check it. Does it now replicate in lung tissue? Or if you've knocked out the right switch, you put it in the off position, then it doesn't replicate in lung tissue anymore. And that's what we did.
Are you able to create just the innards of the virus, just the RNA or DNA, or can you actually create, the capsid and make an entire virion? Or is that not needed at all?
Yeah, in our case, we create the entire virion. So to make a tumor-targeted virus, we made very small but very strategic changes that weakened its ability to replicate in normal cells, but didn't affect its ability to replicate in tumor cells.
Oh, so all right. So you're able to engineer these properties, and then, is the virus selective enough that? All right. Let me back up. How do you know what would make it selectively attack a tumor cell versus a regular cell? Let's say the liver. What's different about the tumor cell that would make it more able to be attacked versus a regular liver cell?
Yeah, so tumor cells have the same receptors that, for example, the GI tract has or the lung tissue has. So they have receptors that recognize the virus. So when we knocked out the particular switches that allowed the virus to replicate it in lung tissue, so it didn't replicate in lung tissue anymore, we had to then go back and recheck it in a whole panel of cancer cells. And we could show that it would replicate very efficiently in a wide range of cancer cells, such as lung cancer, colon cancer, pancreatic, breast, sarcomas, a whole variety of different tumor types.
If this gets into, secret sauce or proprietary stuff, just let me know, no problem. But what would be in common about cancer cells that would not be in common amongst our regular tissue cells?
Yeah, so in cancer cells, they have essentially lost their polarity. So all the cells have a structure to them so that they're organized to be able to communicate with other cells and neighboring cells. So, for example, in the lung tissue or the GI tissue, you want to make secretory proteins that would, for example, give you mucus, but you want that to be secreted into the airways where it can be functioned. So there is a polarity to the cell to make sure that it functions properly and it secretes things normally. In tumor cells, they lose that polarity. And because of that loss of polarity, they lose the ability to selectively replicate or not replicate the virus. And so in tumor cells, because they've lost that normal architecture, they're then susceptible to infection to the virus.
So would a given virus be able to enter into tumor cells and non-tumor cells just as easily, but it's the replication that is hindered, or is it the actual entry?
Yeah, so they can still enter. So the virus can enter a normal cell. It can enter a tumor cell. And that happens all the time. It's just that it can't replicate once it gets in there in a normal cell, but it can replicate, make new infectious particles, and then essentially lyse or kill the tumor cell. because of replication within that tumor cell.
Well, it's very smart. You're harnessing the natural ability of the virus to replicate and lyse a particular cell, one that you don't want, which is great, and not do that to to normal healthy cell. But what happens to a virus that doesn't successfully replicate in the cell? Is there an internal immune system where the cell like recognizes the material and just lets it sit there, or does it chop it up and use it as fuel? Like, what do you think happens to it?
Yeah, so within cells, proteins are always being turned over all the time. So the cell is undergoing metabolism, proteins that are no longer functional or abnormal in some way because they've altered their configuration, get put into the lysosome, which is essentially the digestive garbage dump of the cell. They get broken up into fragments and then recycled. And so viral particles that essentially are ineffective, will go so far in a cell, they'll no longer be of any function, and they'll just get degraded.
Interesting. So, all right. So, is there a residence time? If a virus is not able to replicate in the cell, does it lose out time-wise, and that's why it gets pulled into lysosomes and digested? Is it just that? What is it that makes viruses successful in your estimation in cells, that they're able to replicate, quote unquote, fast enough, or are they able to fend off lysosome activity in the cell?
Yeah, it's very interesting, and it's one of the things that we really tried to understand, or that my laboratory tried to understand, in trying to engineer a cancer-specific virus. And that goes again to that topic we talked about of tropism, that viruses recognize a certain tissue that is the appropriate substrate or the appropriate tissue to replicate in. And so what happens is when the virus encounters a cell with a receptor that it can use, it will get internalized into the cell. And then it tracks along little pathways to get to the nucleus, and it will dock at the nucleus. And along the way, it actually sheds part of its coat. So the virus has the ability to eventually dock at the nuclear membrane and then inject its DNA into the nucleus. And so if that process gets interrupted because it's not in the right environment, those shed proteins get digested, and the unprotected DNA then just gets chewed up by the enzymes that degrade DNA and RNA.
I thought that when a virus enters a cell, the capsid stays outside of the cell membrane and just the interior material goes into the cell. But are you saying that viruses can enter into a cell with the capsid intact, or is there like a subcapsid nuclear envelope for the virus genetic material that's maintained, that sheds once it's inside?
Yeah, different viruses do it differently, but yeah, the virus sheds parts of its coat as it goes into the cell and into the nucleus. I
didn't know that. So some do enter, capsid preserved and all?
Yeah, they can essentially enter into an invagination into the cell, and then enter into the cell that way.
Oh wow, I didn't know that. Huh.
Through docking to the receptor, yeah. So with the adenovirus, it binds to what we call a CAR receptor, which stands for Coxsackie adenoviral receptor. So it's amazing how complicated the life cycle of a virus is. It's essentially not a living organism. It can't make a new viral particle outside of a cell. So it needs to go in and essentially reprogram the cell to be a manufacturing site for viruses. And that's what fascinated me about viruses as a way to treat cancer. If viruses can reprogram a cell to essentially focus all of its attention on replicating a virus, then it can infect a tumor cell and reorganize the tumor cell in ways that destroy a lot of the oncolytic or oncogenic properties of the cell.
Yeah, no, that's really interesting. Huh. So, what are, it sounds like a great solution. How long have you been working on this, and what are the major stumbling blocks to it working?
Yeah, so I started this as part of my graduate work while I was at Stanford, where we were studying how interferon blocks virus replication. And what I discovered was that interferon can block viral replication, in normal cells. And then as I was studying it, I realized as I tested various different normal and tumor cells that interferon would work to block viral replication in normal cells, but it didn't work to block viral replication in many tumor cells. And that led to the idea that tumors were in some way abnormal so that they were highly susceptible to viral infection. As that turned out to be true, then what we needed to do, and the stumbling block was, how do we make a virus that is very safe in normal tissue, but highly lytic and lethal to tumor cells?
Has it been observed clinically that people with certain cancers get sick with a certain viral infection and it actually improves their cancer or gets rid of it?
Yeah, actually. So before the advent of chemotherapy and radiation, the treatment for cancer at the turn of the century was something called Coulis toxins. And Coulis toxins were extracts of various different microorganisms. The idea was that you would cause an acute inflammation, or essentially an infection selectively within the tumor cells. And so there were reports, of the tumors actually shrinking down and even responding completely to Coulis toxins. And so that led to the idea that certain kinds of infections could trigger tumor cell death.
Yeah, because I could see that this would happen naturally. But was there ever a robust process whereby, you could deliberately infect someone with a certain virus that, wouldn't make them horribly sick, but they would get sick, but it would have the benefit of clearing away their cancer?
Yeah, so that's what, so when I was at Stanford, I developed a collaboration with a professor, Frank McCormick. At that time, he had the idea that he could target p53, which is a oncogene with viruses. And he came to Stanford, and we ended up talking about how we could take advantage of my observations about interferon and his observations around p53. So he had already started this company called Onyx Pharmaceuticals, and we started working together to along with the rest of the team at Onyx, to develop viruses that could be used to treat cancer.
So, how far along are you in your effort? Is it, I guess it took years to be able to engineer a virus in the first place. And then, what was your starting point? What kind of viruses? You mentioned adenoviruses. Are there particular ones that, I guess it depends on the type of cancer, but, considering virus X as a backbone, what were your starting points, and, where are you at now with this process?
Yeah, so what we decided to start with was adenovirus. It's a common cold virus. There are millions, if not billions, of infections with adenovirus every year. And it doesn't cause serious complications. So we don't even vaccinate patients for it because it's so common in nature. So the first thing to do was start out with something that was safe. And because it had different serotypes, so the critical finding was that, for example, as I was saying earlier, if we started out with a virus that infected the lung tissue, it didn't infect the rest of the body. It didn't infect, for example, the GI tract or muscles or heart or liver. So pretty much all of the rest of your body is already safe from that virus. All we had to do was alter it so it didn't cause the lung infection and a common cold. But we had to be able to reduce the toxicity from the common cold, but in a way that still allowed it to replicate in cancer cells. So we started out with an adenovirus and engineered it selectively from there.
So how does the FDA view this? Are they really skittish about it, or are you able to fast-track development of something like this? Where are you at in the process? And, is it working in animal models? What's going on?
Yeah, so because it's in an adenovirus, and we have a lot of experience with adenovirus, and we know they're generally safe, the FDA has not had a problem with this, and they particularly like the fact that we could engineer them to take advantage of essentially turning viruses that were trophic for the lungs into viruses that were trophic for tumors. And so I found the FDA to be, very supportive and very encouraging. Of course, we are, like every other company, held to the standard of proving that it's both effective and safe. And so, of course, that's what we're doing. So,
I mean, is it, again, are you at the point where you're testing in animal models, or, how long of a path ahead of you is it? And what conditions in particular, like what cancer are you targeting?
Yeah, so we've completed a large-scale study in a variety of different animal models, and we've started human testing already. So the very first viruses we've engineered to infect tumor cells, but to be engineered as a personalized cancer oncolytic vaccine. So what I mean by that is the virus has been engineered to express the specific mutations that a patient's own cancer has. So the virus will infect the tumor cell and then express those cancer-specific mutant proteins at very high levels. So in the process of killing the cancer cell, it also exposes those cancer-specific antigens to the immune system.
From what I heard, cancers are very heterogeneous, and I would guess as they go along in a person and as they metastasize, they become more and more heterogeneous. Does this therapy work? What if you selectively are just killing certain types of the cancer cells, and other ones are being left untouched because they've differentiated in such a way as they're not affected by this virus?
Yeah, so that's actually one of the problems that we really wanted to address with our approach. And using a genetic approach, we can identify not just one mutation in the cancer, but multiple ones. So the goal would be then that any cancer cell would continue to have its core mutations, and because we have multiple ones, we would be able to pick up many of the different variants. The other thing is that as cancer cells have grown out, if they shift in some way to have a different pattern than what we have in our virus, we can just go back and change the payload and put in those new mutations. So we can adapt our virus to the changes the cancer can throw at us.
Does I guess it's kind of ironic or poetic justice for the cancer. It can't really call on the immune system to help it, because it's evading it. Have you seen that, from what I know, chemo seems to, again, selectively kill off some of the cancer cell lines and others proliferate. Have you seen a reaction to this virus that the cancer now is able to adapt around it, or is it not able to adapt enough?
Yes. So really, cancers haven't been able so far to adapt around the virus. The virus has multiple mechanisms by which it kills cancer cells, or can replicate and turn off machinery that results in cancer cell death. So far, we haven't been able to see ways that cancer cells can change in ways that really protect themselves against the virus. Interestingly enough, cancer cells that are highly resistant to chemotherapy tend to be very sensitive to the oncolytic virus we made.
Well, that's good. Does this need to be personalized? I guess it needs to be personalized as to the cancer, but it needs to be personalized to the individual person as well. Like, do you have to first look at the degree and the character, the heterogeneity of a given person's tumor or tumor burden in order to craft this virus, or can it be used on a population of people that all have a certain type of breast cancer, let's say?
Yes, that's a great question, because we've done both things, because viruses can enter cells and then rearrange the gene expression within a cell. They rearrange it to make it efficient for the viral replication. We can make those viruses perform certain tasks. So, for example, one way that the cancer cells evade the immune system is to overexpress certain proteins that downregulate the immune system. One of them is called TGF-beta. So we've made an adenovirus that goes in, infects cancer cells and kills them, but in the process of killing them, turns off that cloaking mechanism, the immunoprotective TGF-beta. So the cancer cells die, but all the neighboring cancer cells are now exposed to the immune system.
Huh. This is really interesting. I mean, I don't know. I mean, I guess if I were you, I would be freaking out and wanting to get this through as fast as I could. I mean, are there any fast track opportunities, or what's the runway here? How long until this could be used clinically, you think, or approved?
So we've got approval from the FDA to start treatment. So we're just finishing up the manufacturing of the high volume that we can use for clinical material. So we expect to be treating patients this year.
Oh, wow. Well, for which conditions?
Essentially almost any solid tumors, so that it could be colon cancer or rectal cancer or breast cancer or any types of solid tumors that are the cancer cells that this does not seem to work on are leukemias and lymphomas. But other than that, it seems to work on most kind of epithelial solid tumors.
Wow. That's amazing. Huh.
Yeah, we're pretty excited about it. It's been many years to do this really very fine engineering. It's been exciting to kind of figure out how viruses infect normal cells and how they infect tumor cells, and then take advantage of that biology to, create something that selectively kills cancer cells. But we did it, and we were able to alter it in ways that, can give patients a real fighting chance against cancer. Yeah,
that's amazing. Do you think that this will be, so if someone has cancer and they're listening, at what point will their doctor be aware or can be made aware of this possible therapy? When do you think that might be?
There'll be clinical trials that'll be starting later this year. They're going to start at selective centers that are experienced in using gene therapy. So it'll be patients, who are at those institutions or can go to the institutions.
Okay. And then, if you don't mind, just because it's on everyone's minds, any insights that EpicentRx has into the current coronavirus, ways to attack it and kick its butt?
Absolutely. So we're because we can generate an immune response. Our focus is to generate an immune response against cancer cells. As I said, we put in cancer-specific antigens to immunize patients against their own cancer, and we've proven that can work in animal model systems. So making a vaccine against coronavirus is actually relatively simple for us. So we're already in that process.
Oh, okay. So you're one of the companies that is working on that.
Yeah, our specific interest is making sure that we have a vaccine that's safe enough to be used in compromised patients, like cancer patients. And since our vaccine is designed to be used in cancer patients, it can be modified to really be something that would be a tool available not only for the general population, which it could be, but also specifically for patients who are immune compromised in various different ways, including having cancer or the treatment of cancer.
Wow. All right. Well, you're very reserved about it, but this sounds amazing. So I definitely wish you and your company a lot of success, personally, for me, for everyone that needs the help. So, how can people follow up? How can they find out and look at EpicentRx process and progress and keep tabs?
Well, EpicentRx is a small to medium-sized biotech company here in San Diego, and we continue to expand and grow, and our clinical trials will be starting later this year. In fact, we just started last week a treatment. The FDA approved a treatment protocol for one of our other drugs, RX-01, for patients infected with coronavirus.
Okay. Well, is there a website, or what's the best way to find out more? Just Google EpicentRx, or is there a website to go to?
No, there's a website. It's EpicentRx, EpicentRx.com.
Okay. Well, very good. Well, Tony, thanks for coming. It's been a great call. Very interesting.
Hey, thanks for having me on. Appreciate it. And we're very excited to be part of. There are a lot of scientists who are working on cancer and on corona, and we are pretty excited to be part of that group that's trying to make a difference. And we just look forward to helping out as many patients as we can.
Great.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.